Hello students, the topic is biotechnology and its application, and in today's class let us learn how biotechnology is used in the treatment of diabetes. Students, you know that diabetes is a disorder in which the body is unable to produce the hormone insulin. As there is insulin deficiency in the blood, this results in a condition called as hyperglycemia, which can have lot of bad implications for the body and which can be dangerous and life-threatening at times, and can also result in various disorders like heart diseases, strokes, etc. Thus, the only way to improve the longevity and ensure that the person lives a healthy life is that he is given artificial insulin. Now, artificial insulin was previously obtained from other animals like pigs, cattle, etc. These animals were slaughtered and the pancreas was the organ from which the insulin was separated. This insulin was later crystallized and then used in the treatment of diabetes. However, it was seen that the individual who took such type of insulin, which was obtained from other animals like slaughtered pigs or cattle, was a cause of allergy in some people. The body of this individual did not accept this insulin produced in other animals, and therefore it would cause allergic reaction, and hence it was not a good way of treating this condition, that is diabetes, or more correctly diabetes mellitus. The scientists were looking out for a method by which insulin could be produced in its natural form. Thus, biotechnology was used in the production of such type of insulin. So, before we understand how this insulin was produced, let us see what exactly is the molecular nature of this insulin. Now, in mammals, insulin is synthesized as a prohormone. Now, what is the meaning of a prohormone? Prohormone is the immature insulin, and which needs further processing so that it behaves like an active hormone. Now, what is the molecular nature of this prohormone or proinsulin? You can see it in the image shown here. If you carefully observe, students, there are three types of chains which are shown here: the A chain, the B chain, and the C chain. So, the proinsulin has A, B, and C chain. The A chain and the B chain are joined by disulfide bridges. However, the C chain has no such binding with the disulfide bridges. But the proinsulin molecule has all the three chains, that is the A chain, the B chain, and the C chain. Now let us look at the mature insulin. The mature insulin, which is the active insulin, or it is the insulin that actually helps in controlling high sugar levels in blood, if you observe carefully, has only the A chain and the B chain. It does not have the C chain. Thus, we can see here that the proinsulin has A, B, and C chain, whereas the mature insulin has only chain A and chain B. But when the scientists tried to put the gene of human insulin inside a bacteria and get a mature insulin product, they were not successful. This is only because the gene had the information to produce all A, B, and C chain. Thus, the main problem encountered here was to get a mature insulin which does not have the C peptide. Thus, the main challenge for the production of insulin using recombinant DNA technique was getting insulin assembled into the mature form which just has the A and the B peptide. This genetically modified insulin was very successfully produced by an American company called as Eli Lilly. So what the scientists in this company did was, they took the chain A and introduced it in one plasmid. This plasmid was introduced in the bacteria. They took another plasmid to which the chain B was joined. The gene encoding chain B was joined, and this plasmid was introduced in a separate bacteria. Thus, we can see that chain A and chain B were encoded into separate plasmids and which were introduced in two separate bacteria. The bacteria which was used for this purpose is the Escherichia coli bacterium. And then the product which was obtained as a result was collected separately. Like the bacteria in which the plasmid encoding chain A was introduced produced the product A, and the bacteria in which the plasmid encoding chain B was introduced produced the product B. So you can see, your students, that the chain C was not introduced anywhere in the plasmid. So the plasmid was completely devoid of chain C. So the final products had only the chain A and chain B. The chain A and chain B were then joined artificially using disulfide bridging. So we can see, your students, in this image that chain B, represented by the green color, was introduced in a plasmid which was introduced in one type of E. coli. Another chain B was introduced, chain A, was introduced in a plasmid which was introduced in another E. coli. Thus, the two separate E. coli produced chain A and chain B, and these were then joined artificially by using disulfide bridges. This method of producing insulin was known to be highly successful, as this did not have the C peptide and hence it was an active insulin. Besides, all the problems which were caused because of allergy towards the pig and cattle insulin were solved in this case, because this insulin was very much like the human insulin and showed very good activity in dissolving the levels or decreasing the levels of sugar and thus controlling the condition of diabetes mellitus, and therefore was a very effective method for controlling diabetes in such patients. And today, whatever insulin is available in the market is majorly produced by using the above method. Thus, we can see how the production of insulin in bacteria using biotechnological methods has been a very effective method in the treatment of condition like diabetes mellitus. Thank you, students.